Hi, Gaurav. Welcome back. How are you today?
Thank you, Mina. I'm doing well.
So, Gaurav, what are the new amylin calcitonin assays and what do they cover across format and signaling pathways?
It would be right to say the clinical data out from Novo's trial little while ago now, where we have dual-agonist GLP-1 and Amylin as the dual co-agonist that they have put in the clinic, have yielded very positive results. That kind of, I will say, the first place where the entire excitement in the field came in. GLP-1 was already one of the really high impact target that we know for obesity, type 2 diabetes, and other indications, sleep apnea, heart conditions, NASH, MASH. But Amylin, I think, kind of started surging at that time as an interest target for a lot of different organizations to consider. And if you look at amylins, and again, I'm not an expert in that, I can give a very quick background on what these assays are or what these receptors work as. So they are basically based on calcitonin receptor pharmacology. So we have a calcitonin receptor, not to mistaken with calcitonin-like receptor. It's a calcitonin receptor, and then we have a calcitonin-like too. So you take the calcitonin receptor and then pair them with co-receptors called ramps, RAMPs. So when there's a calcitonin receptor paired with a co-receptor, which is a ramp, it makes an amylin receptor. And we have three different amylin receptors, amylin receptor 1, 2, and 3. And very simple to understand, calcitonin pairs with ramp 1 creates amylin 1, pairs with ramp 2 creates amylin 2. And then, of course, ramp 3 creates amylin 3. Out of this 1, 2, and 3, it is kind of more established that amylin 1 and amylin 3 are clinically more significant in bringing the primarily the weight loss is what was looked at in the clinical trial. So coming back to amylins itself, the challenge in the field is everybody is looking for well-characterized assays to start building their own pipelines around amylin. And so cell-based assays already we know have complexity around it. And in this case, you need to nail down the right stoichiometry between your calcitonin receptor and the ramps to get the right pharmacology that looks like an amylin receptor pharmacology. But what is happening is a little far from what the expectation is when you create, a heterologous system such as a cell line and you express two different receptors to it, it's a challenge to get the stoichiometry right. So what is the default form that happens is when you express calcitonin receptor, it is a very well expressed receptor in a cell background, such as CHOs and HEX and other cell backgrounds. It's a very easy receptor to express and usually expresses a very high level. And of course, you co-express the ramps with that. But typically, it's seen what's happening is calcitonin receptor is much highly expressed compared to the ramps. And the antibodies against ramps are not good. We have not identified a really good reliable antibodies for ramps, so that has been a challenge for us to really quantitate absolutely what the stoichiometry looks like. But typically, we have more functional readout that tell us the ramps expression may not be as high or as high as to show a perfect amylin pharmacology when you run these experiments. So what is happening is overwhelming CalCR, which is expressed, and lower levels of ramps. So what you get out of these assays, Yamina, is a calcitonin pharmacology and not amylin pharmacology.
Thank you for that, Gaurav. My next question is about what can researchers now measure and answer about amylin pharmacology that was difficult or impossible before with these tools, especially around the receptor subtypes and bias.
So the challenge is to get the right stoichiometry out from these receptors. So what we have figured out with this one is taking an approach where we are expressing the CalCR receptor using a very low expression promoter. Brings down the expression of CalCR and we can really ramp up, no pun intended, the ramp receptors. So that nails down the right stoichiometry. And once you have that, combination of the right kind of expression profiles for both, now you can come and ask questions like, what is happening to my cyclic AMP signaling? What's beta-arrestin looking like? So that is now a right platform to answer these MOAs, so to speak, or these ways that GPCR is activated using amylin, for example, CalCR, and so many other ligands that are known in the field. So yes, coming to a question where now we have a right platform to address these questions, characterize your molecules, look at amylin pharmacology. The first step is, of course, nailing down these physiologically relevant levels of CalCR and RAMs. So now we have assays built out that can measure cyclic AMP, can measure beta-arrestin. So these are the two main MOAs that you can measure using DiscoverX cell lines for Amylins. And that's what we are putting more effort into it, is to develop more and more assays and also ortholog assays. So not to forget orthologs, we have multiple ortholog assays that we're actively developing, mouse, rat, and other species, because they're also critical. A lot of data is coming out from in vivo systems, and it's important for us to bridge that data into in vitro systems, and that's why we actively develop ortholog assays too. So we have plenty of cell lines that are orthogonal cell lines in addition to humans, and that's what you can do in both arrestin and cyclic AMP formats.
And so my last question is more around how did these assays come to be and what made now the right time to build them? What are some of the challenges in amylin biology that drove the development of these assays, and why are they particularly relevant today? We know that, but I want to hear your perspective.
Right. I think that's a good question, Yamina. I mean, we were thinking we had a portfolio from Amylin assays, but quickly we realized that yes, more is needed. And as I said, to address the gaps in the field, there is an important point that probably people need to kind of think about is the cell background they're using. And that's what we focused on, as you said, the bookends of the things. We focus on the cell backgrounds to identify a really good cell background in which there is very low or negligible ramp expression of its own, because you need to be sure that you don't have endogenous ramps that are going to mess up with the pharmacology. So that is what we invested our time in building those assays. And as you said, the time is right because now there is a lot of interest in the field, and we wanted to make sure that the assays we have, to the clients will be using, have the right pharmacology, right data sets to it, all the molecules that we can get our hands on. We have tested that and really build a portfolio that is well characterized for you to take and really utilize in your own research, knowing that we have put enough time and care in building the right background to it, which has low ramp expressions on the background, as well as putting the stoichiometry right for the CalCR and the ramps. So all those kind of together, we think that we have a really good offering to the market where people are looking to profile, to characterize, and even put an assay in potency testing for these amylin receptors.
Thank you, Gaurav, for the great conversation. I look forward to hearing from you next time. Talk to you soon. Bye-bye.